Welcome to cellular respiration. The first step, glycolysis. Glucose, a 6-carbon sugar, is split into 2 3-carbon sugars. These sugars are then oxidized and are rearranged to form 2 pyruvate molecules. There are 2 phases to glycolysis, the energy investment phase and the energy payoff phase. In the investment phase, 2 ATP per glucose are put in. In the payoff phase, ATP is produced by substrate-level phosphorylation and NAD+ is reduced to NADH. The net yield from glycolysis is 2 ATP and 2 NADH per glucose. Pyruvate oxidation. As pyruvate enters the mitochondria, pyruvate is modified into acetyl-CoA, which enters the Krebs cycle.
Once the acetyl-CoA enters the Krebs cycle, it combines with oxaloacetic acid, forming a 6-carbon molecule of citrate.
The citrate molecule is dehydrated, giving off H2O, then rehydrated.
The molecule then gives off a carbon and a hydrogen, leaving a 5-carbon molecule.
The carbon that broke off forms CO2.
The hydrogen reduces to NAD+ and forms NADH.
FAD+ is also reduced to form FADH.
Next, succinyl-CoA is formed and turned into succinate.
Succinyl-CoA also goes through substrate-level phosphorylation and produces ATP, a molecule similar to ATP.
After a few more reactions, the Krebs cycle starts again.
The net products of Krebs cycle are 8 NADH, 2 FADH2, 2 ATP, and 6 CO2.
4 protein complexes form the electron transport chain. NADH plus H plus enters the I-NADH-Q reductase and is broken down into NAD+. The hydrogen goes through the protein to the other side of the membrane. Electrons are passed through the I-NADH-Q reductase and is carried by ubiquinol to the cytochrome C reductase. FADH2 goes into the succinate dehydrogenase and is turned into FAD+. The electron is then passed on to the cytochrome C and then on to the cytochrome C oxidase. Hydrogen ions and oxygen enter cytochrome C and exit as H2O. H plus goes through cytochrome oxidase. The H pluses in the inner membrane space enter the ATP synthase. It spins and ADP plus phosphate are turned into ATP.
Lactic acid fermentation. Pyruvate is reduced to lactic acid by NADH, which is oxidized to NAD+. Lactic acid fermentation allows glycolysis to continue by ensuring that NADH is returned to its oxidized state, NAD+. Alcoholic fermentation. Pyruvate loses 1 carbon in the form of carbon dioxide to form acetaldehyde, which is reduced to ethyl alcohol by NADH. When acetaldehyde is reduced to ethyl alcohol, NADH becomes NAD+.
Is that it?